Good evening and hello everyone. I'm really sorry. Let me apologize just ahead before I actually start. I've not been able to do the videos I promised some year 11s I was going to do. It's not that I didn't want to do it, but something happened to me on the 1st of April. I had a car accident and I thought it would be something that would go away or be resolved within a week. Today is now the 7th actually. Today is the 7th of May, and the insurance company has still not resolved it. We're actually in a position where I'm seeking legal help now to deal with it. It's meant to be a straightforward thing, but they haven't done it. So it really cost me a lot. I had a pain I had an injury on my left leg and on my left shoulder. I was taken from the whole accident scene to the hospital. So was the other guy, but everybody's alive. I'm grateful to God for that. So it's been hectic. And apart from the accident that I had, I have been so busy at Mind the Gap. Mind the Gap is my own center that I opened January of this year. Word has gone out, and I have had so many referrals. I want to be busy. That was what my desire was, to be able to help a lot more young people who are struggling and who are finding it difficult to get even the basic GCSEs, in math, science, English. That has been my passion, and I wanted to do that. So I'm busy. I have this year a good 5 to 6 lots more young people than I normally have. So I like it. I'm enjoying it, but it's taking a lot of my time. However, that's just why I haven't done it. So today I've managed to start because I found an easy way to prepare slides and then just talk through the slides so I can do a lot more in a shorter time. My One of my daughters was able to show me how to do it. So this particular one is about the required practical. So this is AQA based, but what you will find is that you'll still be able to use this whether you're doing EdXL or OCR. All of these practicals might not be on your paper one, so you need to check. And if you're doing the EdXL or the OCR paper and you feel you need me to look at some other practical that would be on your B1 or that are B1 practical, let me know and I'll talk about those ones as well. So I've looked at the six required practicals for the paper one on the AQA exam board, which they will be taking on the 14th of May. And I've explained everything that would enable you to answer the questions when you do find them as far as the required practicals are concerned. The next one I'll do would be the 6 mark questions. So I want to do 10, a minimum of 10 B1 6 mark questions. So I'll upload that video tomorrow, God willing. All right, so we'll go straight into it. So the first required practical is microscopy. You need to know how to prepare a microscope slide, and you need to know how to answer some questions based on that practical. So the equipment you require will be a small piece of onion, a knife, a white tie, forceps, a microscope slide, a cover slip, a microphone, iodine solution in a dropping bottle, a prepared animal or plant cell. Yeah, prepared plant or animal cell. We normally use the onion cell in biology. And then you need a ruler as well. These are the basic equipment. Obviously, you need the microscope itself. Yeah, the microscope is there for this one. I thought I'll pop a photograph here so you have a bit of idea. So we have the microscope with all these parts. We have the solution and we have the dye that we stain with. And it says here that look for cells with red cytoplasm. Now, when you stain your specimen, what normally happens is that different parts of the cell would absorb the dye differently. Therefore, they would appear different on the slide. That's how you're actually able to identify different parts of what you're looking at. And let me just quickly add that with the microscope, if you're given a microscope, you will not be required to label all the parts. It will just be a few parts that they want you to know how to label, or actually how to use them, more or less, more than labeling them, just labeling for the sake of labeling. So students usually will know the IP is the one you look through, and the magnifying lenses. I usually have three in the one we use, and then you have the stage where you place your microscope prepared slide, and then you have two focusing wheel. You have the fine and the coarse. The coarse is the one that you would use first. If you cannot then see the image clearly, you would then want to use the fine one to focus the inside of what you're looking at, so you can see them really clearly. So as a quick overview, that is the microscope, the eyepiece. The objective lens is the one you change to increase the magnification. There are different strengths. And then you have the stage, you have the coarse focus, and you have the fine focusing as well. Different pictures or different microscopes, when you find them in diagrams, are usually labeled differently. But by and large, the ones I've talked about are the ones that are most important. Now, this question says, describe how you would prepare a slide for viewing under the microscope. Now, you can use a drop-in pipette to put one drop of water onto the microscope slide. Separate one of the thin layers of the onion, peel off a thin layer of epidermal tissue from the inner surface. Then use forceps to put a thin layer onto the drop of water that you have placed on the microscope slide. Number 5, make sure that the layer of onion cell is flat on the slide. Put 2 drops of iodine solution, that will be your stain, onto the onion tissue. Carefully lower a cover slip onto the slide. Do this by 1, placing one edge of the cover slip on the slide, then use the forceps to lower the other edge onto the slide. Now, this is very important. You do this in order to avoid trapping air bubbles. Then there may be some liquid around the edge of the cover slip when you're done. Just use a piece of paper to mop up the excess liquid, and then your slide is ready for viewing under the microscope. So some possible questions that can be asked based on this practical. You cannot see the image at all. You stain it. If you can't see the image, then you probably have forgotten to stain it, or maybe the person that's preparing the slide forgot to add the stain. And then they ask if you can see the image but not too clearly. Then you can use the fine adjustment in order to focus the inner parts of the cell. Bring it into sharp focus, and then you can see it clearly. Sometimes they could say that you can see the inside clearly, but the image is too tiny. I wrote by, it should actually be bought, but the image is too tiny. Then you can select a lens of higher magnification to do that. Then they could ask you a question about resolution. So what is resolution? Is the shortest distance between 2 points on a specimen that can be distinguished by the observer or camera system as separate entities. Long and wordy. So I just thought, you know what, I'll show you 2 images for us to quickly compare. Now, I could say that that is like an image of low resolution. You will see what I mean in a minute. This is actually an image that I found on one of the papers coincidentally as I was going through. But I thought you cannot actually see the stages that these cells are in terms of, are they a prophase or metaphase, or what, you cannot clearly tell. Unlike when you compare with the next picture that I would put up on the next slide. Now, if you look at this, you can clearly tell that F is at metaphase. They are lining up in the middle. And if you look at H is telophase because they're splitting into two. And for I, this is the cytoplasm forming around each daughter cell, which means that is cytokinesis. You can just tell clearly what's happening at these stages. So these are Google images I pulled up, and this can clearly tell you what the differences between an image has been looked at and has been seen through a high resolution microscope and the one that hasn't. So that was why I thought I'll put them for the purpose of comparing the two. So that's resolution. So if you're looking at two circles through high resolution, it will clearly show you that there are two circles or two round structures. But if it's poor resolution, if it's low resolution, it might merge them into one. You might think you're looking at just one circle or some kind of shape that's not distinct. Okay. So they might also ask you why an electron microscope is better than a light microscope. Well, an electron microscope works by a beam of electrons, and so the image will be naturally sharper. Apart from that, it has a higher resolution and enables the specimen to be seen in greater detail. So if you're looking at really tiny images, you can be sure that with the electron microscope, you'll be able to see every tiny detail. So that makes an electron microscope better. It's more modern, it's better technology, isn't it, than a light microscope. You could be given a picture that looks a bit like this, an image of some sort of a prepared slide, an onion slide, or some kind of slide with what I want to highlight are the dark spots. So they could ask you what the thick dark spots are. I have heard students say before that is the nucleus or labeled some part of the cell. It's not. That is air bubble trapped. So when you're using the cover slit, when we say you need to be careful and lower it with the forceps really gently, it's so you don't trap air bubbles. If you trap, if you manage it, or if for instance you trap air bubbles when you're putting the cover slit, that's the way they will appear. Very simple answer to that question. Is trapped air bubbles is not part of the cell. I thought that's really important for me to add to that. And another thing that came to my mind whilst doing this voiceover or recording, was I remember that if you say you want to work out, if they say, oh, the eyepiece is at times 10 magnification, and the one of the objective lenses you are using is times 40. For you to get the total of those two, how they magnify your object is that 10 times 40. So you have to multiply the two together. That's another question that you could be asked. And remember, you're allowed to use a calculator, so don't freak out. Some of you just see anything that looks like math and you freak out straight away. Please don't do that. Pull the calculator and do it. Many of you can solve that, but just use the calculator to multiply the magnification, the lens, the size of the lens of the eyepiece, and the particular lens that you're using on the objective lens that you've selected. Multiply the two to get that answer. So that said, though, the magnification formula is image over object. You need to know how to rearrange this particular formula. If you're giving the magnification already and you're giving the object size, you can rearrange it to work out your image size. In that case, it would be the magnification multiplied by the object size that you're giving to give it the image size. Or if you're giving both the image and the object size, and then you can work out your magnification the way this formula is written. And then another thing you need to remember is the conversion. If you want to convert from millimeter to micrometer, or then onto nanometer, or then onto picometer, at each point you multiply by 1,000. If you're going down the other way, you divide by 1,000. You could be asked to present your answer in a standard form. I thought of it just now, but if and I didn't add it when I was preparing the PowerPoint slide earlier on. However, it's easy. I do have a video on on my channel here where I talked last year that I did about the science in math. I did cover how to work out the standard form. They will find a way to test you on standard form and the little pieces, bits and pieces of math in science. Um, if you're the kind of person who freaks out about maths, please don't freak out here. There's a chunk of questions that will test your numeracy ability in science, and it's all of those things like standard from significant figures, decimal places, percentages, and those are the little things that they ask you. I have a video that covers that. Do check it out. And just as an example here for the conversion, if you're given a measurement as 2.1 mm and you need to work it out as micrometer, you multiply by 1,000, you get 2,100. And if that's handy for anyone out there, you remember you're allowed to use a calculator. So don't freak out. There's no big deal about the maths in science. Just remember to use your calculator if that's something that would give you some comfort. Anything that would help you just stay calm and write your exam paper is what my desire is for you. So yes, we will carry on. Now you could be given a diagram and something similar to this, and you'll be asked to calculate the length or the diameter of the nucleus. Now, from what you can see here, the diameter of the cell is 76 micrometer. So what I would say, if you use your ruler to measure the diameter across the nucleus, then you work out how many lots of that ruler lies along the diameter of the entire cell. So if you know, when I did mine, it was like 4 lots of it could lie along the 76 micrometers. So I did 76 divided by 4, and I got 19 micrometers. So the width, the average width of that nucleus is about 19 micrometer. Then the required practical number 2 is growing bacteria. The materials that you need include the agar gel, the petri dish with, excuse me, a separate lid, the little paper disc, a selection of disinfectants and bacteria. What I did when I was putting this together, I picked bits and pieces of information from different places. So you might be asked to, it might be that someone's researching into the strength of different antibiotics. It's just something that can kill bacteria, and then you want to compare 2, 3, or 4 of them. And then some students wonder what are paper discs. When you hole puncture your paper, the little round papers that come out are your paper disc. That's what we actually use in the lab to do it. So these are the basic materials that you need for that. The method, you set up a Bunsen burner to kill any bacteria in the air around where you're working. Then only work with the petri dish under the Bunsen burner flame. And then using a sterile pipette, take out some amount in mil of bacteria from a sample. So the bacteria that has been prepared. Then squirt the sample onto the agar. Dip the a spreader into some ethanol and burn it off to kill any bacteria. So because you don't want to infect your agar jelly environment. Then you spread the bacteria around the plate evenly. Then you seal the plate in 4 places using tape. You don't seal it all around, so there is no, actually, you don't want to make it completely airtight like that. So, where you want to make it airtight, but you don't want a situation where there will be a problem when you're to interrupt with your growth of your bacteria. Then you put a label on it indicating what it is and when it was spread. Incubate this plate for 3 days upside down. That upside down is very important to avoid condensation. Because if you do it the other way around, the lid that you cover it with could gather some liquid, and then it could be condensed. It could condense into water and then contaminate the result. Then you sterilize used equipment. That's not serialize, it's sterilize. You sterilize all the used equipment. You sterilize it because you don't want it to spread bacteria everywhere and in the places you don't want them. A student was asked to grow an uncontaminated culture of microbes in agar jelly. The teacher gave the student a method. The equipment and method are shown below. I've just talked about the method, by the way. These are the equipment. The petri dish with the lid, the tape, Bunsen burner, the inoculating loop, and culture of microorganisms. Some notes just to cover what we've talked about. One of the discs was dipped in water. Why? This is the control. This is the control. This is the one that doesn't have any disinfectant. So this is your control. Then the independent variable here is a different disinfectant. And then the dependent variable is the width of the clear area around each disinfectant. Because when you leave your disc inside the bacterial medium that you have grown, after some time, you will notice that there are clear areas around the disinfectant because you would have dipped the disinfectant in your paper disc into the disinfectant. When you then put it on the agar jelly, you discover that there is clear area around your paper disc, showing that some bacteria have been killed off by the disinfectant or the antibiotic that you're trying to check its potency, how good it is at killing bacteria. If you look at this diagram to the right, you will see that there are different widths around each paper disc. The paper disc with antibiotic is the one that's there, and then this is the petri dish, and then the area around each paper disc is the area that you have no bacteria growing. It's cleared off all the bacteria. Now, what is the purpose of the agar jelly in this experiment? And below the diagram or the image, I answer that the agar jelly is for nutrients for the bacteria to grow. And then another question, by the right, I said it's a result of one of such experiments. Which disinfectant was most active on the bacteria? Very clearly, it was number 3 because it has the widest area around it that the bacteria has been cleared off. You could be asked to explain why the disc size must be kept the same for each disinfectant. So you can get, to make it effective, because a wider disc would absorb more disinfectant. So you want to have accurate results that are comparable so that, that becomes your constant, not another variable that's affecting the results that you get. Then give one possible source of error in this investigation. If the bacteria has not been evenly spread over the agar plate, then there could be a difference in outcome that's not a reflection of how efficient the disinfectant or the antibiotic actually is. Then if there is contamination from your hair or your fingers on the agar plate, that could highly affect the result that you get at the end. Then they said here, the student decided to calculate the area of the region with no bacteria using the formula to calculate the area of the circle. This would not give an accurate measurement of the effectiveness of the antibiotics. The reason why is because the clear regions are not exact circles. They are not perfect circles, so you cannot use pi squared to just calculate the region. So you have to actually use a ruler to measure it to know how wide it is for you to work out the width, the diameter of each area around each disc. Required practical number 3, osmosis. You need a potato chip, a core borer or potato chipper. You need a vegetable stick cutter, a ruler, 10 centimeters cube measuring cylinder, labels, 3 boiling tubes, a test tube rack, paper towels, a sharp knife, a white tile, a range of sugar solutions, distilled water, and a top pan balance. These are the equipment that you need for osmosis. Osmosis is an exciting one because when I was a little girl, if I just pop a story here, you know how I'm like, I like to tell stories. When I was a little girl, my first contact with osmosis was my brother. He had done it in school. I was still in primary school, and he came home and he went, I am going to teach you some science. And I'm like, okay, bring it on. And then in Africa, we have the pawpaw fruit. The pawpaw fruit is like, I don't know what I'm going to use to compare it. Here, I don't know, it's like the god the gourd plant is similar. I don't know if that's a good comparison, but the purple fruit or the papaya is very common in Africa. So he went to the bar garden and got one from the little shrub-like tree and cut it in half. Now, when you cut the papaya tree, it has the inside has got seeds, but it's like you have a lot of space in between. It's hollow on the inside. He then put some salt solution into it and then put the papaya into a bowl of water and covered it. The following day, I was so amazed. The water that was inside the papaya had risen, and I'm like, how did that happen? How did that happen? And he went, that is science. I'm like, what sort of science? He went, that is osmosis. So I never forgot when I got to school, my teacher was teaching me osmosis in secondary school. I just remembered that practical and I was really excited. So osmosis is actually movement of water particles from a region of high concentration to a low concentration through a semi-permeable membrane. Is on the next slide. Not this one, the one after this. So this one is the one you have the list of the diagram of the equipment. I just thought I'll pop the diagram of the equipment because some students, remember this is lastminute.com. I'm just like getting you to do bits and pieces, just rounding off your revision for you. If you've not been revising, don't panic. You have not written the paper yet. Rather than panic and fail, just pick up bits and pieces of the information. And if you've been through school, all of these revisions should help you remember lots of things. Okay, so you need the potato, you need the distilled water in that bottle you can squirt, you need the core borer, you need your sugar solution, the test tube rag, the scale, the measuring cylinder, there's some blotting paper. This is actually a white tile, and then you have the test tube and your forceps. That is a scalpel, right? For cutting the potato chip. You use a core borer and then you can use a scalpel to trim it off. You measure it on the scale. So let's look. I think the definition is not on the next on the next one, or the method. I think is the method that's on the next slide. So use a core borer to cut 3 potato cylinders of the same diameter. Trim the cylinders so that they all have the same length, about 3 centimeters long. Accurately measure and record the length and mass of each potato cylinder. Then measure 10 centimeters cube of the 0.5 mole sugar solution and put into the first boiling tube. Label the boiling tube as 0.5 m sugar. Measure 10 centimeters cube of 0.25 m and sugar solution and put into the second boiling tube. Label this tube as 0.25 m. And although these steps that I found from the AQA website, although it's just 2, it's advisable that you actually have up to 3 different solutions, and you have one that is just water, so that you can have a range of results. Because I'm thinking on the exam paper, they could give you a very simple or simplified method of any of these required practical. That happened last year for making salts. I think it was the list of the steps involved, and you had to correct the errors. So you need to know how to carry out each step of the method, so that if you're given a step that does not that's not completely correct, you can then work out the errors and state how you would improve them. So I use this information to put together a table of results so that we can see how typical results look like and how we can use them to answer questions. And obviously, I did uh, include some definitions of some keywords that surround the osmosis practicals. We'll look at that in the slides that follow. So you measure 10 centimeters cube of the distilled water and put into the third boiling tube. Label boiling tube as water. Add 1 potato cylinder to each boiling tube. Make sure you know the length and mass of each potato cylinder in each boiling tube. Record the lengths and masses of each potato cylinder. Leave the potato cylinders in the boiling tube overnight in the test tube rack. Remove the cylinders from the boiling tubes and carefully blot them dry with paper towels. you can then remeasure the length and the masses of each. So this is like an overview of the method that you need. What I did, I put together is secondary data, because when you do your practical, you can take a secondary data to compare and for the analysis of possible outcome. On the one that's going to be on your paper, it's not going to be an actual one, or it could be an actual one, but it's not the one you've done. So it's a secondary data which you can use to analyze or test your hypothesis. Hypothesis being the actual science or the question you have asked about the science that you're trying to work out if it is the case or not. So we go look at what more we've got on osmosis practical. I think this is the third one. We've got three more. So let's get going. Anyway. So here's a result table I put together. Very important. Very important. Notice that I have put the initial mass, the final mass, the difference, and then the percentage change in mass. That is very key. Students will know the difference in mass, they will know the change, but to work out the percentage, and percentage is one of the maths in science, so you need to be able to work out the percentage change. I'll show. So this tells you how it works. So the difference, yeah, the difference between the start weight and the start mass and the final mass. You divide that by the initial mass, multiply by 100. You get the percentage change in mass. Now I pulled up this graph so that I can use it to explain isotonic point. The isotonic point is where the amount of sugar or the concentration of sugar in the potato is the same as is outside in the potato, where the test tube that the potato is in. So most of the time they can ask you to use this graph to estimate the concentration of the potato chip. In which case, if this is the graph we're looking at, it would be at 0.3. It's not minus. This will just be putting a little dash there. It's 0.3 moles. Now that is the case because that is the case because that is where the graph actually hits the x-axis of the graph. So that's the point where the amount of water that's going out of the potato equals the amount of water that's going into the potato. So either flow is either way is the same. So that's how you can estimate the average concentration or give an estimate of the concentration of the sugar concentration of the potato chip. I hope that is clear. Please remember these points about the osmosis practical, the definition which I already gave you. Remember semi-permeable membrane. I had to highlight that one. Sometimes they will say partially permeable or selectively permeable. They mean one and the same thing. It means that a membrane that would allow certain things to pass through and stop certain things from passing through. In this case, the potato membrane would allow water molecules to pass through. It would not allow the sugar to pass through. So it's selectively permeable. Where we see osmosis in nature is absorption of water by roots from the soil. This is by osmosis. And then diffusion, I had to give that definition, pop it there, is the movement of particles from a high concentration to a low concentration. This is where how gas exchange happens in the lungs, and it is by diffusion. The third one is active transport. I always talk about the three of them together. My students know I always do them together because you need to know these to hand. They are very simple explanations and simple scientific ideas to remember to have to hand, because you can just get another 5 marks from these simple definitions. Sometimes it's not that students don't know what they're asked to do, they don't remember, or they can't remember how they link together. So I love to do the link, create the link. I'm talking about osmosis practical, but I'm referring to diffusion, I'm referring to active transport because they go hand in hand. There is a link between the three of them. You got it? Good. Now, active transport, this is the movement of particles from low to high concentration with the use of energy. I highlighted low and high because in this situation, particles are moving against the concentration gradient. It's like going uphill. So energy is needed to push the particles from low to high concentrations. Where you find this active transport is in mineral ion uptake by roots from the soil. Now, there was a question I was looking at with one of my students yesterday, and she was a bit confused. It says you have a lot more mineral ions in the root hair cells than you have in the soil. So what's going to happen? Or and then the question went ahead to say, but in the root hair cell you have a lot of mitochondria. Mitochondria is where respiration happens. What they were looking for was for you to explain active transport. That for that to happen, active transport is the movement system of moving the ions which are few in the soil, a low concentration area, to the root, which is high concentration. And the reason why you have a lot of mitochondria is the site for respiration. And respiration is the process of using oxygen to break down glucose to release energy. Because energy is needed in active transport, you need energy to push these molecules up against the concentration gradient. That is why the root hair cells have a huge supply of mitochondria, because they need the ATP, they need the energy that is really byproduct of aerobic respiration. I was able to explain to her. Then we went on to look at anaerobic respiration. I like to link things. I don't want to veer off on that. So another place we find it in is in digestion. When you eat your food, the amylase start to break it. Amylase enzymes start to work on your food from the mouth. Eventually, when your food gets to the small intestine, you have glucose. Now that glucose is pocket. I call glucose little little packets of energy. They have energy in them. Then you need fire, the oxygen, to break open these little pockets of energy and release the energy you need. So that is respiration. And in active transport, respiration energy is needed, and it comes from the mitochondria respiring. In anaerobic respiration, you don't have enough oxygen. So there is an oxygen there. There is a buildup of lactic acid. If care is not taken, it can cause cramp. And that's why sometimes sports people, they suffer that kind of cramps in their muscle. Burning sensation in their muscle is active. It's as a result of lactic acid. I thought I'll just chip those ones in. Science is so exciting. And when I'm doing revision, I get so excited because I want people to remember. I want my students to remember everything that they've learned from year 9, and I don't want them to forget anything. So when they go in their exam, they can get their grades. You know what? You're not going to pass. Remember, keep revising. I won't say that all day. I want to say that. Okay, I'll go to the next slide quickly. The independent variable in the osmosis practical, the independent variables are the different sugar concentrations and of the solution, and then the dependent variable is the percentage change in mass. The constant, which is the one you measure, the constant variables are the volume of sugar solution used and how long they stand for. They are left to stand for. The risk here could be cutting your hand with knife. Be careful. Sugar solution could get in your eye, wear eye goggles. Because when you look at a risk factor, you have to explain how you can prevent that risk from happening or protect yourself against such a risk. When you bring the chips out of the solution, you mop them with paper towel. They could ask you, why do you do that? It's so that you remove excess solution. Require practical 4 food test. As a matter of fact, I said to my students, I am not going to do this. I will overflog that because I had a video on this last year, and it was one of the 6 marker on the triple paper, I think. So the food test, simply put, as you have to test for starch, protein, sugars, or glucose and fat. You need to know how to test for these different types of food categories or classes. I just thought I'd put it in form of a table, a very simplified table. For your food, starch, you crush the food and add iodine solution. There should be a color change, blue-black, if starch is present. And then for the glucose, you take the food and add some Benedict solution. There should be a brick red or orange color change, or there should be a range of color changes depending on how much sugar is present. And then for proteins, you take some egg white or the food that contains protein, add some Biuret solution. There should be lilac or purple change if protein is indeed present. Then for the emulsion test, take some fatty food, crush and add some ethanol to it and mix it. Then you can pour some of this solution into water in a test tube. If for you to get an emulsion, for this to be properly done, there should be a milky color formed at the top. A milky color formed at the top. This is your emulsion in the water. This is how you test for fats. And that's food test that you need it. If you need more information, you can just ask me which aspect you need information with. I just thought I'll do it like a really quick overview. If we go into lots and lots of details, students might not remember. This is just a quick, as quick as I can make it, review of food tests. Some notes, though, that I could think of that are associated with food test or digestion and all that while I was putting this together. I just thought, bile is produced in the liver and stored in the gallbladder. Remember that? And then the food, what's the function of bile? Why is bile so important? The food coming from the stomach during digestion that goes into the small intestine is actually acidic. So if you don't want it to be acidic while it gets to the small intestine, the bile is used to neutralize the acidic food before it enters the small intestine, which is alkaline. Bile also emulsifies fats, very important. It breaks it into smaller pieces for it to be easily digested by the enzyme lipase. Breaking it down this way increases the surface area for faster digestion. And I said to myself, I'll find a diagram and pop it there to explain how bile emulsifies fats. There you go. That's the photograph. Yeah, that's the image. So you have a fat globule, and then that's bile salt does that to it. Emulsifies the fat drop, and then as it's like this in little chips, little bits and pieces, it's easy for it to be broken down really, really quickly. The surface area is increased, therefore it is a lot faster to break down. Required practical 5 is the enzyme. It's enzymes and pH. pH is how acidic or alkaline a substance is. Enzymes are proteins. They break down food quickly, but to do that, they do not take part. They do not take part in the reaction themselves. Enzymes are biological catalysts. They are reaction specific. The enzyme that breaks down carbohydrates will not break down proteins. That's what it means by saying that they are reaction specific. They work best at an optimum temperature and pH. Beyond this, they become denatured. This means that their active sites get changed, so they don't die. So then say, they get destroyed. I'm like, no, enzymes don't get destroyed; they get denatured. And by denatured, we mean that they have changed their active site. So if it was rounded, it becomes maybe square or something like that. So that gives you kind of a picture. So the substrate can no longer fit snugly onto the active site. Therefore, it can no longer act on the substrate. Say it was breaking down starch. If starch molecule can no longer fit onto the changed active site of the enzyme, then the enzyme cannot catalyze that reaction. The enzyme cannot break down starch into glucose. So and higher temperatures be above 40 clearly denatures the enzymes in our body, and pH also affects enzyme. Some enzymes work best in acidic and some work best in alkaline environment. So this particular practical in the AQA is about checking how enzymes are affected by different pH levels. The list of equipment include test tubes, a test tube rack, water bath, spotting ties, 5 centimeters cube measuring cylinder, syringes, a stop clock, starch solution, amylase solution, buffered solutions covering a range of pH, each with a labeled syringe and plastic pipette, and then iodine solution and syringes. Now, let me just quickly say that if you test if you test starch with iodine solution, it will be blue-black. By the time it changes to orange, it only means one thing: the starch has been broken down into glucose. Yeah, so you need to remember that to hand. Method. Oh, I should have spread this out, but well, let's see how. I'll just talk you through it. Place 1 drop of iodine solution into each depression on the spotting tile. Place labeled test tubes containing the buffered solutions, amylase solution, and starch solutions into the water bath. Allow the solutions to reach 30 degrees Celsius, because at very low temperatures, the enzymes cannot function. Add 2 centimeters cube of one of the buffered solutions to a test tube. Use a syringe to place 2 centimeters cube of amylase into the buffered pH solution. Use another syringe to add 2 centimeters cube of starch to the amylase/buffer solution. Immediately start the stop clock and leave it on throughout the test. After 10 seconds, remove 1 drop of the mixture with a glass rod. Place this drop on the first depression of the spotting tile with the iodine solution. Use the glass rod to remove 1 drop of the mixture every 10 seconds. Put each drop onto the iodine solution in the next depression on the spotting tile. Continue until the iodine solution and the amylase/buffer/starch solution remain orange. If they stay orange, then it has become glucose. It's no longer starch. Repeat the procedure with solutions of other pH. Then you can compare and see at what point. So maybe 1 pH, the blue-black color changed to orange after 30 seconds. Maybe another one, it took maybe the 11th tile, in which case it was nearly the end of the experiment. So pH A obviously is a better the enzyme work best at pH A than at pH B. I've put some pictures to just further explain it in the slides that follow. So I decided to pop this image here for you to just see as a way of reminding yourself. This is not, I didn't do this. I just thought I'll pop this here for you to see what we mean. So these are your iodine solution already put there. When you then start to use the glass rod to drop, you put a drop every 10 seconds into these depressions, then you will be able to observe the color change. When you put the first one, it would turn blue-black, and then it would gradually change as according to how much starch is present or not present. This is a graph that could help you see what the outcome could be. This is not the one for that experiment. Again, I just want to use this to show you. And some of these graphs could be on your paper. You should be able to clearly see that this one was, this reaction is clearer than this one. This one did not go there that high. And so you can be asked to compare the two like the reaction of how amylase breaks down the mode, and how pH affects how amylase breaks down mode and arsenic, another strain of microorganism. This is just a way to show what I was trying to do with this particular slide or diagram is to show you what the graph could look like of pH and amylase breaking down starch or breaking down a material. Now, I also want to use this to show you temperature, because on some exam board they don't do the pH, they do the temperature. So you do, because temperature also affects enzymes. And like I said before, at certain temperature they work best. That's their optimum temperature. And then at certain temperature they become denatured. They no longer work. And you can see from here, if you look at when it was 60 degrees Celsius, there was no digestion at all. And at 0 degrees Celsius, there was no digestion. If you then come to 20 and then 30, you can see the difference between that. From 10 to 20, 30 to 40, you can clearly see. By the time it got to 50, it took much longer before you could see any change at all. So that's how you can then use all of these to plot all of this information to plot a graph. That would be a bell, shape graph. Iodine turns blue-black if starch is present. When it is no longer blue-black, no starch is present and glucose has been produced. That's the interpretation of the color changes when you do this experiment. A graph such as this could be drawn, and this graph explains what's happening at different places. Because they could give you a graph like this on your exam paper and ask you should explain what's happening at the different points that they have marked. So from 10 degrees Celsius to 40 degrees Celsius, the rate at which starch is digested to sugar increases with temperature. As temperature increases, it increases as well. The rate increases. At 40 degrees Celsius, the rate of reaction reaches a maximum. This is the optimum temperature for the enzyme. And above 40 degrees Celsius, the rate at which starch is digested increases as the decreases, as the enzyme becomes denatured. Required practical 6 is the photosynthesis. I did a lot more about this on my videos last year. It's still the same exam board I'm doing this year, AQA. And if you refer to those videos, I have over 40 videos. Not all of them are GCSE revision, but many of them are GCSE revision videos. Please check. And if there's any particular one you want me to repost, I can repost them or maybe tell you how to get them. But I think a lot of people are finding those videos anyway. So just get there and use these videos to help you. So a boiling tube, a freshly cut 10 centimeters piece of pond with a light source, a ruler, a test tube rack, a stopwatch, 0.2% solution of sodium hydrogen carbonate solution, a glass rod. Then those are the equipment that you need. Then the steps include set up a test tube rack containing a boiling tube at a distance of 10 centimeters away from the light source, like your lamp, for example. Fill the boiling tube with the sodium hydrogen carbonate solution. Put a piece of pondweed into the boiling tube with the cut end at the top. Gently push the pondweed down with the glass rod to ensure it's fully immersed, isn't it? So that every part of it is respiring. Leave the boiling tube for 5 minutes. Start the stopwatch and count the number of bubbles produced in 1 minute. Repeat the count twice more, and then you can then use the data to calculate the mean number of bubbles per minute. I emphasize this in the next one as well. That's so important. Repeat steps 1 to 6 with the test tube rack and boiling tube at a distance of 20 centimeters, 30 centimeters, and 40 centimeters from the light source. Those are your independent variables, the one you are changing. Questions would be typical questions here would be about how to improve the experiment in order to get more accurate result. Maybe repeat 3 times. If it was not already said, you should repeat 3 times and take an average. Sometimes this experiment is done with alga balls to ensure that the exact number of alga are photosynthesizing to ensure you get a more accurate result than the pondweed. And then human errors such as miscounting could occur. And I'm thinking if you use like a gas syringe to collect the gas, that could be a way of getting a more accurate result if we could work around that. The risk here would be the sodium carbonate hydrogen solution. It is slightly toxic, and large doses could be corrosive to the gastrointestinal tract. So any part of your belly, your digestive system could be affected if ingested. So be sure not to ingest. Wear goggles and wash hands after experiment. I always say to students, we all have different types of sensitive skin. Some of us don't have, some people have very sensitive skin. So be very careful and then some students put their hands in their mouth without thinking. Please, after experiment, wash your hands thoroughly so you don't get bits and pieces that should not be in your mouth into your mouth because it could affect your body. You don't want to put any toxic thing into your system. And so there you have it. There are 6 required practicals on paper 1, AQA biology paper 1 that you'll be taking on the 14th of May. And just keep revising. I wish you the very best in your revision and on towards your exam. I'll try my very best to put up the 6-mark question about same time tomorrow, because it will take me like a bit of time during the day to finalize the questions. It's a bit difficult to pick which ones, but I'm just going to pick a selection so I can cover different aspects of the B1 topics that you've covered. So keep revising and keeping hope up. Don't give up. Every little bit of revision is important. It's an added, it's a positive. Please keep revising until you take the paper. Don't give up. Don't give up. Just keep revising, and then it will all be well. See you in my next video, and God bless you.